It's Mark Gardner and Caroline Lowe from AMG today, and we're going to take you through 3 topics. Caroline's going to start with freely available databases with applications in drug design. This is really building on the session last time, which focused on Chembl, and Caroline's going to talk a little bit about Chembl, but mostly about some other databases of interest and relevance to medicinal chemists. And then I'm going to talk about a simple Knime script looking at comparing compound collections. Really the start of this was James Duffy at MMV, who was asking for a simple way of evaluating collections of compounds that they might be interested in screening and looking at how they complemented what they had already screened. And the third one is a DataWarrior item on SAR, and it's really, I guess, just sharing something that I personally find very useful as a way of looking at SAR of reasonable sized data sets, a few hundred molecules. So I will close the poll and share that with everybody, and then we will get started with Caroline's item.
Ooh, right.
Hopefully you can see the results there. So I'll now hide that and turn the session over to Caroline.
Okay. Can you all see my screen?
They won't be able to say yes or no, but I can, so
Okay.
I think everybody's
Can you see
fine.
my screen, Mark?
I certainly
Okay.
can.
Lovely. Right, so I have put together a slide set on databases that are useful in medicinal chemistry. I know that Anna Galton gave a quite a detailed talk on Chembl at the last webinar, and if any of you missed that, there is a link to the recording of that on the MMV website, as there is for all the other talks that have taken place in this session. So, but there are other questions that you might want to know and get information from. For example, are there any protein structures known about my protein or closely related proteins? And how do I tell which residues form a binding site for the compound, and where are they in relation to the small molecule ligand? There are databases that can give you information, probably more like pointers, to questions about what the biological target for your hit compound might be if you're screening in a phenotypic assay. There are databases that give information about selectivity, whether your compound binds to other proteins, whether it's known in the literature, whether the molecule that you've identified as a real hit or is one of these compounds which interferes with binding assays and known as pains. You might want to be able to assemble a virtual screening data set, perhaps one that is targeted towards your particular profile, the profile that you require. And you might need to know about the sort of hydrogen bonding interactions. There's a big discussion about whether intramolecular hydrogen bond formation is an important parameter to be considering. And you can get access to the small molecule crystal data that can give you information about this as well. So here's a collection of my favorite databases, ones that I use daily throughout the course of my work, and they are all public domain databases, by which I mean they are free to access. I'm not concerned in this with freedom to modify the database, and none of these databases would come under that category. I also need to point out that these are my personal views as a user of these resources, and it's only since I started putting this slide set together that I've had a chance to delve a little into the background of some of these. It's quite clear that there is considerable overlap between some of these, for example, well, in particular between Chembl and the PDB. But there are now numerous links between each and every one of these databases. If you pull up a structure on the Protein Data Bank, you will find links out to all of these databases that you can readily get to. Now, I didn't want to go into Kemble in any great detail, but there are some very nice features that Anna alluded to, but I thought you should know about in a little bit more depth. So, for example, there is a set of some 5,800 compounds which have been deposited by AstraZeneca on the Kemble database. The way to find these from Kemble is to search the term ADMET as a target. The alternative way, and somewhat quicker way, is probably just to Google AstraZeneca DMPK set and Kemble. And that is actually the easiest way of getting to this. And then you will find that there is a large data set with data on log-Ds, solubilities, pKa's, and some in vitro plasma binding and intrinsic clearance information. The data set is not complete, so there aren't 5,800 results for every single compound, and in actual fact it averages out to about 2.2 activities per compound. Almost everything has got a log-D value, and about a third of the compounds have got kinetic solubilities. But once we start getting down to microsomal clearance values, this is a much smaller proportion of the database, although the numbers of records here are probably larger than anything else you'll find out on the web. So these have all been run under the same conditions, and references are given to the assays used, and this is a useful resource for model building and validation. For example, if you wanted to test how good a particular algorithm was at predicting one of these activities. Finding these specific data sets is quite easy if you click on the activity source filter, which is on the right hand side of the menu bar, and it will give you a full listing of everything that's there in order of the number of records here. So by far the biggest proportion of Kemble records come straight from the scientific literature, but there are quite a few data sets here which are relevant to the global health community, and most of these you can get to from the menu bar on the left hand side, which is a lot easier than trying to get there via Google. There was quite a nice feature called the ADME SAR Safari, but that appears to be down at the moment for essential maintenance, which I assume means it's being extensively revamped since it's been down for about the past 2 weeks. So I won't go into that, and we'll leave Kemble at this moment and move on to PubChem, which is a subject that we haven't really discussed at these meetings yet. It is by far the biggest data set, publicly available data set there. There are screening results for many novel targets, and here you can see that there are results for some 91 million compounds tested in 1.2 million bioassays. And 33% of the active bioassay records are not on Kemble. So I thought I'd have a look at what the criteria were for passing information between these two big databases. And it turns out that all the literature results from Kemble are in PubChem, but only a subset of confirmatory and panel assays which have dose-response endpoints are put into Kemble. The biggest source of records in PubChem that aren't in Kemble are those from the NIAID, which contains data from screening in HIV, opportunistic infection assays, and TB. And Chris Southern has done an ongoing analysis of the overlap between Kemble and some of the other databases. This is not between Kemble and PubChem. It's between Kemble, Drug Bank, the Human Metabolome Database, and the Therapeutics Targets Database. So this is a four-way Venn diagram. So you can see in 2010 there were some 600,000 compounds in Kemble, but between 2010 and 2013 this number roughly doubled. But in actual fact the number of records that overlap for all four databases was relatively small. It was 115 and it's now gone up to just over 1,000. I recommend, if you want to know more about the informatics, behind this, go and have a look at Chris's blog, which is down here, and this information from the 2013 study is published in this paper here. I should also point out that Kemble has probably doubled in size since 2013, and the latest release is considerably larger than this. So we need a way of comparing how much data there is on both of these databases. So from this point onwards, I'm going to use resveratrol as an example. It is, of course, the compound that you find in red grapes and hence in red wine, and has been mooted as the fountain of life by numerous publications. So it's been screened many times in multiple bioassays. And I'm very pleased to note that my favourite red wine, which is Chateau Neuf du Pape, has almost 4 times the levels of resveratrol than certain other wines. So I shall keep drinking that. If I look up resveratrol on Kemble, I get 1,400 assay results concerning tests in 215 protein targets, and these are nicely broken down in these pie charts here. So you can see what types of results they were. We have functional results, binding results, a fair proportion of ADME results, and some physicochemical properties here. We have results from enzyme assays, membrane receptor assays, and very much smaller numbers on things like ion channels. You can also break this down from the output, which tells you where these records come from. The vast majority of them are from the scientific literature and have been curated by Kemble. About a quarter of those are from PubChem bioassays, and a fair size of them are from this drug matrix database, which is a toxicology database. So remember that number, and we'll go and do exactly the same search in PubChem. So this is the new front end of PubChem that you'll see if you go onto the PubChem website. It's in beta testing at the moment. And PubChem finds 3,000 records versus the 1,500 for 1,400 for Kemble. It does have this very nice target-based analysis where you can clearly see what sort of bioactivities you have. So we have 589 of those 3,000 records show activities for resveratrol, whereas 1,500 of those records show that there's no activity. Now this might indeed be useful information if you're looking for selectivity of your particular compound, so don't dismiss this type of information. We can see that there are 5 records where resveratrol is extremely potent, and these come top of the list. They're all in anti-proliferative assays, and here are their activities in micromolar. It tells you what sort of bioassays they were and the breakdown of activities active in inactive compounds and unspecified. And you can easily drill down to the types of targets the compounds were tested in, and the types of bioassays. I think this is a very nice layout for drilling down through assays that is an equal of Kemble's. Of course, I find that PubChem has some annoying features. One that's always bothered me is that as a medicinal chemist, it takes 3 clicks to reach the structure activity. So I thought I'd go through this exercise with the new beta interface. So you can search the compound database, and that brings you up to this panel here. You still need to click on the structure tab, and once you've got to the structure tab, you need to click on this little icon here, which is not in fact labelled and has no tooltip, in order to bring up the structure drawing interface. So we're still at the level of needing 3 clicks to reach the structure activity, structure editor. And which I think is something that PubChem might like to address perhaps in future releases. An alternative way of looking at the data that you get in these databases is to use the Open Facts Explorer. Now this is quite a new piece of software. It's basically a data aggregator, and it's pulling data from Kemble as well as ChemSpider, SwissProt database, and a number of other systems. That it's not pulling data from PubChem. but it does contain PubChem records. It contains those which are already in Chembl. They also provide an API and workflows to answer specific questions which are documented on their website. So these are Knime and Pipeline Pilot protocols, but I have to say that this is not a terribly easy system to set up. So if you're a new user, I suggest you use the Explorer, which you can find at this address here. And this is the type of results you get. So we're down to the 17. We now got 1,700 records versus 1,400 from Chembl and 3,000 from PubChem. But it's a nice clean clean interface, and it's just a different way of looking at the data. Which brings me on to the binding DB, which does have some nice additional features in addition to the data records for each of the compounds. It was launched in 2000, and there were major updates in 2007 and most recently in 2015. By major updates, the 2015 update was a very large update, and there didn't seem to be an awful lot that actually went on in the intervening years from 2007. But the database now contains some 1.25 million records from 549,000 compounds. It's manually created, so there's a lot of information on the assays that the compounds were tested in, and it also contains some ITC calorimetry data. There is no phenotypic data here. All the data values are either measured experimental Ki IC50 values of that time. But there are a couple of nice additional features that I thought you might like to know about. And these, so first of all, let's show you what happens if you just search for resveratrol. So there's a much smaller number of hits. There are 102 hits that you get. This was a simple text search, and this is one record from the output. This is the one that came up top. It's quite a busy interface. The information you really want is here. It tells you that it has a Ki of 88 nanomolar in this assay here, which is inhibition of human quinone reductase 2. It contains links out to the protein data bank, so you can get an X-ray crystal structure of that interaction. But each record contains the same structure of resveratrol and links to the same set of databases. So it's quite a busy interface. It's quite difficult to screen through all 102 hits. The really nice features are these ones here. So there are both web interfaces and Knime protocols that allow you to ask the question, what is the likely target for my compound? So this is very useful if you are dealing with results from a phenotypic assay. So you can draw your structure in, ask it if there are any records with biological activities in. Here it says there's one record, and this is the record that it pulls up. So you can see the similarity between the two. And there are Knime protocols for this that you can download, both for the tell me which target my compound hits and tell me if there are any known ligands for my target, which is the direct inverse of that. And if you're interested in those, have a look at this paper here. Which brings me to the daddy of all databases, as far as I'm concerned. This is the Protein Data Bank, which has become the global repository of all published protein and nucleic acid structures. The majority of them are X-ray structures, and a smaller proportion are NMR structures. And there is a growing level of cryo-electron microscopy such structures, which are now at sufficient resolution to be useful to a molecular modeler. It's always worth actually searching the PDB, even if you can't find literature references to your structure, to your protein, simply because it also contains the structures from the SGC, which aren't always associated with the publication. So there are currently 14,000 structures and over 1,000 ligands, which you won't find in any other way. You can search by name, sequence, ligand and function, and there are many online tools that you need to visualize them. There are some notes here on using the structures. The take-home message is, if you're going to use these in any kind of modeling exercise, then please use some kind of checking software before you use them in anger. So let's have a look. This is a search for Zika compounds, small molecules that interact with proteins of the Zika virus. And it's pulled up the first of these, which was published in July in Science. Here's a cartoon of the structure, and there are links straight underneath to viewers that you might want to use. Alternatively, you can download the file and use a standalone package, something like Isara, which is free for everybody to use. And if you're not familiar with that package, there's a tutorial available on the MMB website in the computational chemistry section. The first thing most people want to know is, what is my compound actually interacting with? So here's the Zika compound. It's a boronate ester, and these 2D pose view images are available for all ligand-bound structures. There's a growing proportion of these. They now form 36% of the database. This number has increased significantly, mainly due to the methodology that they use now for generating high-resolution proteins, because carrying out the crystallization in the presence of a ligand makes it easier to crystallize the structure. So here we have all the residues which interact with our small molecule ligand. They're all labeled in terms of the residue ID. Here's ASP 1129 and the chain ID, so you know whether it's binding between two chains or just in one specific binding site. And there is also a 3D viewer that can give you a picture of your ligand with the protein side chains around that you can turn around. I can't do this on this. This is a 2D. This is just a PowerPoint screenshot. You can search the protein database by ligand. That's simply under the search menu on the home page. And clicking this button here will take you through to the structure drawer, which you then draw your compound in the usual manner and search for that. And there are also direct mappings to drug bank. And if I run a search for resveratrol, I find that there are 21 protein structures which all contain resveratrol, and I can analyze the binding sites of those, and I can look for similarities and differences that I might want to exploit, perhaps if I'm looking for selectivity. On the topic of modeling, if I want to build virtual screening data sets, then the place to go is the Zinc database. This is provided by the Shoket Irwin Labs at UCSF and is now in its 15th iteration. If you are going to Google Zinc, you will be taken to version 12 of the database rather than version 15, so it's worth Googling Zinc 15 from the outset. It now contains the catalogues of over 260 vendors, so that's over 100 million purchasable compounds. Now, there has been a recent update on purchasability within the last month, I think, actually. But it has to be said that by purchasable, it's probably not as efficient as doing a screen on e-molecules. So if you're just after a set of compounds to buy, you might consider just using the e-molecule interface, which is also free to use. But Zinc is exceptionally useful because you can create customized data sets for drug-like, lead-like, and fragment-type compounds, and you can now specify these types quite tightly. It also has some interesting facts on its homepage. For example, there are 150 substances in current clinical trials with a Paynes pattern in them. There are also 60 which are known to aggregate, and aggregation is known to cause false positives in vitro binding assays. So, and some of these questions are well worth investigating just to see what the answers are. Zinc is also very useful if you want to carry out an analog by catalogue search. So if I search for the substructure of resveratrol, I have 194 compounds that I can go out and buy, and I can quickly expand the SAR around by examining the chemical space around this simple core in a very easy manner. Which brings us on to the final section. What sort of 3D information is known about the small molecules? There are some records in PubChem which have links to the CCDC, the Cambridge Crystallographic Database. And this is really useful information because the crystallization of the small molecule itself tells you whether how the compound forms hydrogen bonds between unique monomers of each compound, and the presence of any intramolecular hydrogen bonds. So here's a familiar molecule to the global health community, and by clicking on the link from PubChem, I can get directly to this very nice interface. If I pull down unit cell under this packing menu here, I get this view here, which I can turn around in 3 dimensions and look at these types of interactions. Now, most of the I couldn't find how many records on PubChem actually had CCD records, either by looking on either provider's database. So I'm not sure how many there actually are, but it's been brought to our attention that there is also an open database which is hosted in Lithuania. It's called the Crystallographic Open Database, and it contains not only models of organic compounds, but also everything up to minerals. Its one exclusion is the biopolymers, which are covered by the PDB, and there are some 366,000 records there. Only a quarter of these are organic compounds, and you can't get the same type of view that you get from the CCDC. It shows you the size of the unit cell, and it shows you a picture of the molecule, but it doesn't give you information about the crystal packing. And you can now access this data directly from DataWarrior. It's one of the database searches which is inbuilt into the latest version. You need to have downloaded the current version in order to do this. You can just search for resveratrol, and it searches the organic subset of the COD, which was a snapshot and contains 90,000 compounds. It gives you the crystal data here, but it doesn't give you the picture that you see on the COD, but you can link out to that. So that's covered all the databases that I use in my work. I probably use them all the time, really, and I'm sure that some of you on the line are probably more expert than I am. And if anybody has any comments that they'd like to make, I'll turn the floor over to you now. I just put this last slide up here. This is a date for your diary. On the 12th of October, the SCI, of which I'm a member, and the RSC's Chemical Information and Computer Applications Group, which Chris Swain is a member of, and I know he's online, are running a meeting at the Imperial War Museum in Duxford. And if any of you are interested, the list of confirmed speakers is down here, and you can see that some of them, particularly Isabel Giraud, have given presentations to this forum, and you'll be able to meet them in person. If you're interested, see the SCI website for booking details. Thank you.
Great.
Mark, have you got any questions?
No, I don't think I do. In view of time, I'm going to crack on, but please, as Caroline said, please do contribute your own thoughts via the questions interface or via email afterwards. As Caroline said, we're by no means have a monopoly on thoughts on useful databases or any of the other material that we're presenting, so it would be great to get your thoughts and ideas and your own best favorites and favorite ways of using these things. If you could submit them via the questions, we'll distribute them, or by email we'll add them as well. So that would be fantastic. I'm going to change over now to the second topic, file comparison workflow in Knime. So this, as I mentioned earlier, was based on a request from James Duffy at MMV, and we've ended up with 2 use cases, which is not surprising because these things are quite related. So the principal use case, the first, the reason for the workflow was to be able to do a diversity selection. So if you have already screened a substantial number of compounds and somebody's saying, what about this file, you might want to know, how that second file, which we're calling a query file, complements the first one, first collection of compounds, which we're calling reference compounds. So that's the first use case, but also just by doing the same kind of similarity comparison, you're going to be able to find analogues of actives. So you might be interested in screening additional compounds that might have additional utility for you as compounds to test to get similar activities. So that's the use cases. In terms of the functionality, so you're putting in two files, basically a reference file, which is the file that you know about, and a query file, the file that you're considering testing compounds from. And then you also get two outputs. The top of these is essentially just the query file annotated with various things like similarity to the reference file and some physchem properties and that kind of thing, which we have a template for viewing in DataWarrior, but of course you can go and view it in any SAR visualization tool that you'd like to. And also this produces a summary report, and mostly this is summary statistics associated with the query file, simple things like the numbers of rows, the numbers of unique structures, the exact matches with the reference file, mean similarity of nearest neighbors and mean molecular weight, that kind of thing, and also a couple of lines associated with the reference file. So that's just a summary of the inputs, basically. In a bit more detail, just because it did cause a little bit of confusion early on in terms of the reference file. As I mentioned, that is your file containing the compounds you know about. The query file is the test file that you're considering acquiring either all of or part of. So just a little bit of what the workflow does. Primarily, it's about analyzing similarity between the two collections. Schematically, it very obviously identifies query compounds which are identical to reference compounds. So I've just represented those by overlap there, so that's a very simple thing. It's also looking at similarity. So it gives a similarity of each query compound to up to 4 similar reference compounds. So you get a column which tells you the similarity of the nearest neighbor for A and B, and in this schematic you can see that the similarity of those query compounds to their nearest reference compounds is pretty similar. But what we wanted to do was add in a bit more information, because if you're considering a file that might complement what you have already, you might decide that A is more valuable to you for that purpose than B, because B is surrounded by several compounds which you've already tested, so you have several neighbors to B, whereas A only has one near neighbor, and you might decide that therefore A is more value to you than B. And so basically the protocol calculates a column which we call predicted underscore value, and that is higher for A than for B. So in that use case of looking at compounds that complement what you've got already, you could use that column pred underscore value to select compounds to give you diversity over what you've screened already. I won't go into detail in the calculation, but it's in the slide deck. And if anybody's got any questions about that, if there's time later, we can talk about it, or if not, just ping me an email. So just a little bit more on the workflow. It removes salts. It deduplicates both files. Actually, I don't calculate canonical smiles just for time purposes. So just to be aware of that, if anybody's sort of technically minded, I kind of felt that most input files would be deduplicated, or at least contain the same way of calculating smiles. So actually all I do is deduplicate on the input smiles, technically, but anyway, that's what's happening. And then it picks a single compound ID per smiles as well, if more than one are present. You can optionally count Merkle frames in both files. That adds a little bit of overhead of time, which is why I've made it optional, because the intent is that you should be able to use this with pretty large files if you want to. It then also calculates some properties of the query file molecules, such as log P and rule of 5 fails and that kind of thing, and then a similarity matrix of the query compounds to the reference compounds. Again, I mentioned that we produce some summary data, so there's two files. This is one of them, this is the summary output, and so you're getting things like structure counts in the query file, some similarity information associated with exact matches and similarity information, and if you have included this option, it counts the Mirco frameworks, both the absolute number and the percentage of unique Mirco frames, the number of unique Mirco frames and the percentage that are unique associated with all of them, and the mean molecular weight, mean log p, that kind of thing, so some file properties, and then a couple of notes about the reference file as well. In terms of visualizing the output, this is a template which is produced for DataWarrior, so you can, there's instructions at the cheat sheet at the end that I'll show you in a minute, so you can apply this template to visualize the output, and you can see that you just see the properties of the query compound. So the rows are query compounds, all the query compounds are present in the output file. If there are nearest neighbors, then they are present on the right hand side of this file, so you can see where there are two structures together. The left-hand one of those is the query compound, the right-hand one is the most similar neighbor in the reference file, if there is one above 0.3 using the similarity method that we're using here. And that predicted value thing that I mentioned is a column in the middle there, and you can see that it's 1 for that top compound because there's no similar neighbor. It's quite high for the next one down because there's only one similar neighbor, and it's not a particularly similar neighbor at that. Going down to much lower scores if you have several highly similar neighbors. So the fourth one down has several fairly similar compounds associated with it, so it's a lower predicted value. And the fifth one down is a predicted value of 0 because there's an identical compound in the reference collection, and it's assumed that you don't want to screen the same compound twice. Also in the template you get, actually there's a macro to calculate some binned properties that you can then visualize in the template with things like molecular weight and S-log P and things like that. And then, as I mentioned, if you can optionally select this to calculate the Mirco frames of both sets of compounds, and then they too are presented here with, again, the Mirco frame of the input molecule on the left and the Mirco frame of the reference compound on the right, and you get information about the number of times that Mirco frame appears in each collection. So that can give you some information about the diversity, I guess, of the collections. So on this section, I'm just going to conclude with a couple of suggestions about how you might use this for each use case. So for the first use case, where you're selecting for the novelty of chemical matter, I guess this predicted value has been put there because the aim would be to calculate or to select compounds with a high predicted value. So you might want to set an appropriate cutoff, just by eyeballing, ordering them by predicted value and eyeballing the query compounds and their nearest neighbors, and deciding, where your tolerance is as to how near your near neighbors need to be and how many of them you want. So that's kind of a use dependent thing, but have a look at that. One thing to note, and this is common to any sort of method of comparing one file with another, compounds in the query file that are unusual are likely to score high on this predicted value feature, and particularly if the reference file is large. That means it's probably a collection of a lot of compounds that are reasonably drug-like that you've looked at before. That predicted value score will select for you lots of unusual compounds that don't match anything in the reference file. That might be what you want, but you probably want to look at the compounds with high predicted value and check property distributions and apply filters and things like that as required. So then, having filtered down to the properties and the predicted value you like, my personal preference is always to eyeball a random subset of compounds at this point. I have included, in fact, a random number generator that the workflow puts in, so that all you've got to do, having selected a subset you think you might like, is order by the random number, look at the first 50 to 100, and the acid test basically is, do you want to screen them? If the answer is no, then you may need to repeat your selection criterion, or have another look at the query file and think about whether it's the right one for you. The Merco frame count, if you've selected to do that, now the statistics associated with this will obviously apply only initially to the unfiltered query file, so it'll do things like count the number of times that particular Merco frame appears in the query file. That'll give you some impression of diversity within that file. It's also worth noting that DataWarrior can create a diverse subset as well, or cluster the file, or cluster the thing that you've filtered down to. So if you want some, DataWarrior has a help within it. You can have a look at the options for clustering and creating diverse subsets. Just one word here, if you want to manipulate just a subset in DataWarrior, an easy way to do it is to select the subset and then create essentially a new file within DataWarrior. You could export that file then and repeat the calculations using the workflow in Knime if you wanted to, and that would give you now a set of new statistics associated with the Merco frame counts in the query file, maybe the one that you're considering now using. So that's suggestions associated with selecting novel chemical matter. And of course, if you've got several files to consider, you could repeat the analysis for each of the potential query files and compare some of the summary statistics and things like that. And maybe that would be a way to narrow down to the file or two that you were most interested in doing some detailed work on. In terms of the other use case, this is hit expansion. So within the template for DataWarrior, there is a view there which is like the one on the right-hand side. So the most likely scenario here is that your reference file is fairly small and has got one or a few structures of things that you want to find analogs to. Just a note that if you haven't done this too often, my personal view is if you have a series that you're trying to find analogs to, it's always worth putting in a few structures, not just one, because the different representations of that series may pick up subtly different similar compounds. Anyway, having done that and put in the compounds you're interested in your reference file and the large file you're searching in the query file, you could obviously filter out the compounds where the similarity is 1, in other words, the exact matches, because presumably you don't want to screen them again. And you could also remove compounds with no similarity to the reference compounds, and then you could look at the similarity to each reference ID plot, which is on the right-hand side here. So the most similar compounds are going to be at the top to each of your reference compounds, which you could display the ID or even the structure if you don't have too many on the x-axis, and then go ahead and apply the property filters, etc. as before. So to finish up this section, just to note that in this slide deck there is a cheat sheet that should contain everything you need. I'm going to show you the workflow in a minute, which looks like the stuff in the middle. And there are links to things like DataWarrior if you haven't downloaded that before, the macro and the template that I mentioned earlier, and some online help for using Knime and DataWarrior if you haven't used those before. So I'm just going to show you the workflow because I wanted to show a couple of features. Firstly, when you select the file you're interested in, if you're looking at large files, you probably want to start by limiting the number of rows that are actually input, which you do with that advanced button and limit rows, and then you can say, well, just read, however many, 1,000's probably fine. DataWarrior, Knime looks through the entire file to check that basically that the columns are all formatted correctly. You can, just hit quick scan, and it sort of stops doing that if you're confident that the files are of reasonable format. And so that's that part. The the other element is this renaming. So the workflow operates with just an ID and a smile string from each file. Depending on what your file contains, you might need to rename these things. You will need to get a hold of a smiles representation. So for example, if your compound IDs are called molregno, you'll need to click on this to change that so that you change it to reference ID. And another thing to note here is that you will need to make sure that DataWarrior Knime thinks that this is a string. It's going to treat it as a string. Okay, so now when it goes to the rest of the workflow, ref ID will be the thing that is this one, and you'd remove the Kemble ID. Okay, and you can do the same thing with the smiles string if you need to. So that's renaming the columns. So you need to make sure that you've got everything sorted out inside the yellow boxes. The last one is this optional one, which is whether you want to add the Mirko frames or not, and that's just a 1 if you do and a 0 if you don't in this box here. Okay, so I'm going to end that piece there. Let me just check if there's any questions about that. All right. I'm going to move on now to look at the final topic of the 3, which is SAR by R group analysis in DataWarrior. And this is really a sort of personal preference, if you like, as to how I like to look at SAR. One of the ways I like to look at SAR, obviously I like looking at compound versus lipophilicity plots and that kind of thing. But one of the things I like to do is I like to have all of the compounds on one scatter plot, and I like to be able to look at rows and columns where I know that in this case the columns all share one particular R group feature, perhaps the core or a different R group, and the rows are identical apart from that single R group feature. Okay, and then a nice feature of DataWarrior is you can duplicate that view and sync them together so you can look at a different property on another panel. So in this case I'm looking at microsomal half-life on the right-hand panel, and the potency against the parasitic worm on the left-hand panel. Now this is a project I'm involved with, so I'm not showing you the structures of that one, unfortunately, so I have downloaded a PDE5 example instead. So we're going to go through that in a minute, and from that sort of dummy data set, which we'll download from Kemble in a second, I just wanted to show you the kind of thing I mean. So here we've done an R group analysis of the PDE5 data set. This compound is sildenafil, and to the left of it on the same row you can see a different core where all we've done is taken the nitrogen from this position and moved it to that position. So the rest of the substitution is the same because it's on the same row. And on this at this position it's a different core. Again, we've done another nitrogen walk. This time we've taken it from this position and put it in the bridgehead position. Okay, so there's another row where those 3 cores are also compared, and if I just isolate those 6 compounds a minute, there are instructions on how to do that here, which I won't go into to get this visualization in DataWarrior, but basically all those 6 values come from the same reference here, and actually DataWarrior gives you a way to link out to the references from the Kemble data. You can see that this scaffold looks as though it might be more potent. These are nanomolar potencies against human PDE5, and if we go back to the original plot, then you could say, okay, well, so how about making some of these analogs, because in this sildenafil core these substitutions seem to give greater potency. So if we substitute for what looks like a more potent core, maybe we'd find some good compounds here. And many of you may know this is actually the core present in vardenafil, which is very, very close to sildenafil apart from the core and I think a ethyl group here instead of methyl. Oh, look, I thought I'd replaced the inverted nitrogens. Sorry about that. Anyway, yeah, and vardenafil is used clinically as a lower dose, so maybe there's something in this core potency thing. So there are more slides here about how to do some of these steps, but what I'm going to try and do now in the 10 minutes remaining is actually demonstrate it, which is always a dangerous thing to do. This is one I've sort of prepared earlier just in case. So first of all, we're going to start by searching the Chembl database from DataWarrior, which is a really nice feature. You can start to navigate down the protein family tree by selecting from this hierarchy, and I'm going to focus on PDE5, and having done that I'm also going to focus on PDE5A. Okay, so we're now going to do that search live. I'll come back to this substructure query and the way I formatted that in a minute when we do the R group deconvolution. So we've now got a new instance of DataWarrior, so let's boldly get rid of the other one, and we've now got 282 records. I'm going to get rid of a few that have 0. Ooh, that's interesting. Oh, there we go. A value for, as you know, this in Chembl, this is the potency column, and sometimes you need to sort of clean up the data. Obviously your own data I'm sure would be much cleaner than this, so I'm going to delete those rows, and there are also some with very high values for potency which I'm also going to clean up just for the purposes of this demonstration. So let's go down to greater than 10,000 and we'll delete those rows as well. Okay, so the next thing we want to do in order to get those views that I've been showing you is to consult my crib sheet. And so DataWarrior actually provides some ways of doing SAR analysis, and you know there's some help associated with these. There's some activity cliff stuff, so there's some quite interesting things here, but I'm not going to explore all of those now. The automatic SAR analysis looks like a good place to start, and you know that will give you something that looks like this, and in some cases you can see that the core is present as you know, and then we've got R groups off the core, and that looks great. In other cases it's different parts of the molecule that have been broken down, and that isn't really quite how I want to view this. So I'm going to delete the rows, the columns that we've just created, which includes the core fragment and each of these R groups, and we're going to manually do the same kind of thing. So now we're going to do a core-based SAR analysis, which is where you're going to do this manually. So let me just show you that essentially I've built up the core. It's the same query that I used to query the database in the first place. I just want to show you one feature of this which is quite nice, because if you put in variable atoms here, so these we've got any atomic number for all of those 5 atoms, and if you double click now, having selected those bonds on one of the bonds, you can see that you can vary the nature of the bonds as well. So what that effectively means is when you do the R group deconvolution, and sometimes you get a couple of cases, in this case where there's symmetry that you might need to deal with in the real case, but what that means is when you do that you get the scaffolds separated out here, which is nice. So now in order to generate, there's one more step in order to generate the views that I've been showing. So what we've been doing is on the x-axis we're looking at one feature of the molecule, in this case the core, and we're keeping that constant up the y-axis. So the y-axis basically needs now to contain a concatenation, or the way I do it, concatenation of the smile strings for all of the other R groups. And that is possible to do in DataWarrior using, basically you go to chemistry, add smiles, and you can add a smile string for the structure, which is what you'd expect, but you can also add smile strings for any of the other structures here. I'm not going to do that right now. I'm going to show you a macro which will do it for you, which is here. So these macros are just text files. You can record them in DataWarrior and then you can export them and edit them, which is a really nice feature. So the core element of the macro is between this task name and task end of task signifier. So what we're doing here is adding a smiles code. We're going to call the column smi_scaf, and we're doing it for the structure column scaffold, and then we're repeating that for each of the R groups, R1 to R5. Okay, so that chunk is adding smile strings. The next chunk is adding a column which is a concatenation of everything apart from the scaffold in this case. So we're going to call it less_scaf. So it's the smile strings apart from the scaffold, and the one below it is less_R1, so it includes the scaffold smile string but not R1. Okay, so all of that is in a macro, and if I then read in that macro, I can then run the macro, and you can see columns getting added, and I'll show you what they look like. So we've got the smile strings for each of the R groups and then the concatenated versions there, and that's fine. So that would give us a way to visualize, say, R2 against less_R2, but we want to order these axes in a way that's going to make sense to the visualization. And if you remember Isabelle Giraud's presentation, you can do that by right-clicking on any of the columns and then set category custom order. And you can, in fact, put these in an order yourself manually if you want to, or you can do it according to properties by, for example, row count. We'll group all those columns which have with the most rows at one end, which could be a nice way to do it. In this case, I'm going to use the lowest of that potency value, and then I just hit sort categories and OK, and we generate a new order of that particular column for visualizing. Again, we can do that with a macro as well. I'm not going to show you this one, but I'll just import it, and that's order the axes and then apply the macro. And then the final step is to apply the template, and the template has the DWAT ending, and if the magic happens, then we've got a visualization now of the each of these tabs contains a visualization of the R group on the x-axis with everything else on the y-axis. As I mentioned, you can also create a you can duplicate that view, create another version of it, move it to the side, and then we can color that by a different property. Now, there aren't many additional properties in this one, so I would use solubility, for example, but what you can do, I'll just show you this very quickly, is synchronize this view to the scaffold one, so that if we move about in this view, then we also move about in the one on the right, so that you can home in. If you imagine this one colored by a different property, you can home in on the SAR that you're most interested in. So I apologize that that's gone right to the wire. There is information on how to do all of this in the slides as well. please do contribute your own thoughts on how to do some of these things through the questions interface or through emails afterwards. I know we're at time. If anybody has any burning questions, please raise your hand now. Otherwise, we will conclude. Is anybody wanting to say anything? Okay. Thank you very much indeed. Been a bit of a rush this week. Our next session, I think, will be looking at using predictive models, and Caroline's organising that one for October, so look out for something in your emails for that, and you will get a link to the recordings, and if you haven't downloaded them, through the handout section, the PowerPoint slides, which contain links to everything you need, templates, macros, workflows, all of that. Thank you very much.